Hi, my name is Alex Tenalia, and I'm going to be discussing the article called The DMRT3 Gatekeeper Mutation Affects Performance of Nordic and Standardbred Trotters. This is just a little bit of information about the authors of the article and when it was published in August of 2014. For years, scientists believe that there is a direct correlation with genetics and outstanding locomotion in the trotting performance of standardbred and Nordic trotting horses. To test this hypothesis, scientists measured this mutation in the DMRT3 gene in these horses to determine if this gene was the cause of the improvements. Today I'm going to go over what the mutation is, the scientists' hypothesis, the scientific study of Nordic trotters and standardbred trotters and how it was determined, and the results and conclusions of this study. The mutation of the DMRT3 gene causes a premature stop codon and is expected to result in a truncated DMRT3 protein. It is a change from cytosine C to adenine A and a permissive for performing alternate gaits such as pace or ambling gaits, and has a highly significant positive effect on harness racing performance in standardbreds. Further evidence for a critical role of DMRT3 in coordination of limb movement was obtained by characterization of locomotion and spinal cord function in a DMRT3 knockout mouse. As you can see in this little diagram here I have on the screen, all the genes surrounding the DMRT3 gene, such as the DMRTB1, C2, and so on, they all depend on the locomotion of the DMRT3 gene that helps with the coordination of the limbs. The scientists' hypothesis was if the DMRT3 gene is mutated in a Nordic trotter or standardbred trotting horse, then there would be a direct correlation in the horse's performance. In a blind study on trotting techniques in Nordic trotters, a professional trainer divided a group of 73 horses into 3 different subcategories according to their trotting ability. These categories were horses with a smooth, symmetrical, even beat trot without rhythmical disruption, a group of horses with difficulty sustaining an even beat trot at high speed, and a category of horses with a tendency to pace while trotting but could not trot at a very high speed once they were trained. Within these groups, the trainer compared the DMRT3 genotypes with the classification of the horses as well. After grouping the horses into these 3 categories, the scientists compared the genotype classes between them. The horses that were classified with a clean trot mostly had genotypes of CA. Horses classified into the group that had difficulty keeping an even beat trot at high speed were predominantly CC homozygotes, and the group of horses that had performed at a pace more often were AA. There was also a group of horses that were misclassified, but did not have a large impact on the statistics. The trainer's classification scale also indicated that AA and CA horses had a better harness racing capacity than CC horses. The table shows the varieties between the 3 groups of horses and how the genotypes correlate to each classification of gait. The equation P equals 2.1 times 10 to the negative 8th is the equation for calculating the EBV for trotting technique. There is also thought that the age of the animal could be a factor in determining when the gene mutation occurs. This next graph that I'm about to show you describes the relationship found between different age groups of standardbred horses and the mutant DMRT3 allele. The mutation of the DMRT3 gene in standardbred horses has an extreme correlation between age groups and affects the ability in trotting horses' performances. According to the article, the consistency of the mutant allele has been constant at a frequency of about 98% since the 1950s in standardbreds, while for the Nordic trotters, the frequency reached over 30% in 1970 and has been relatively stable since then. Although the mutation of the DMRT3 gene and age affect the performance of these athletes, like any other athlete, so do surrounding conditions such as weather, surface type, diet, and many more. Here we have the graph that I was just describing. As you can see, between 1950 and the 2000s, the CT has been an increase and relatively stable, and up top, the frequency of A allele in the standardbreds are relatively frequent throughout. The hypothesis that the mutation of the DMRT3 gene in Nordic Trotters and standardbred trotting horses causes differences in the athletic ability of the horses was tested by scientists and found that the differing genotypes of the mutation directly affected the horse's ability and performance. After discussing what the DMRT3 gene mutation is, as well as the process of determining its athletic effects, scientists are now able to understand how this mutation changes the horse's athletic abilities. Because of this advancement, horses will now be competing among the absolute best.